Hi everybody, this is a quick recording to go over the couple of slides that we couldn't finish yesterday. So the last mechanism for post-translational gene expression regulation in eukaryotes that I wanted to share with you is the one that is mediated by ubiquitin and the proteosome. So the way this works is as follows. Let's say we have any protein in the cell, and the cell wants to get rid of that protein quickly. It may not be enough for the cell to simply stop the expression of the protein. Many proteins in cells are long-lived and they could hang around for a really long time, and the cell may want to get rid of that protein and the protein function as quickly as it can. So in many cases what it will do is it will use the ubiquitinylation of specific target proteins and the ensuing proteosome mediated degradation. So here on this slide we see how certain proteins are targets for ubiquitinylation. That is a process by which molecules of ubiquitin that are shown down here in their actual atomic or protein structure form get attached chemically via ATP consumption to target proteins. That is a chemical reaction that is mediated, like many other chemical reactions, by enzymes. These are ubiquitinating enzymes that recognize the target protein and know where in those target proteins to attach a series of ubiquitin molecules in a chain. This is what we call normally polyubiquitinylation of a target protein. What that does to the protein is it marks it for a huge machine that exists inside of eukaryotic cells called the proteosome. Just like we have the spliceosome, a big machine for splicing, the replisome, a big machine for DNA replication, we have the proteosome. This is a big machine for protein destruction. What happens is the proteosome will recognize those proteins that have been polyubiquitinated, and it will take them on one end of the proteosome, and inside there is a bunch of proteolytic enzymes that will chop up that marked protein into its component amino acids. So think of it as a shredder, a protein shredder that takes these marked proteins and shreds them up into individual amino acids. Now the proteosome also has a way to distinguish these ubiquitin molecules from everything else in the protein, so it will actually spare those ubiquitin molecules, take them apart and recycle them so that they can go and ubiquitinylate the next target protein. Once again, this is a mechanism that eukaryotic cells use to get rid of proteins that they no longer need, and they need to get rid of them quickly. Sometimes just stop the transcription and translation of a given protein won't get rid of the protein that already exists, and this is what the cells do to get rid of those. Okay, that's the last mechanism for post-translational gene expression regulation that I wanted to share with you. I hope you enjoyed this video, and I'll see you next time.